This is Chapter 17, dealing with immunization and immune testing. Starting with immunization, there's two broad categories for administering or providing immunity against different types of infection. There's active immunization and passive immunotherapy. Active is when the antigen is administered, so the patient is going to mount their own response involving the adaptive immune response. So therefore, they will ultimately produce antibodies. They have been exposed to the antigen, that foreign substance. This usually will provide long-term protection for the individual. Passive immunotherapy is when, for various reasons, the person is not able to be exposed to the actual antigen. So therefore, they are getting immunity. How? Because someone else's antibodies are being transferred to this individual. Those antibodies will provide protection short term. There will be no memory response with it. It's just it will protect them for right now, but not down the road. Just very brief history of immunization. Chinese are actually the ones who started, not calling it immunization, but they essentially were doing that when they noticed that children who had had smallpox, they did not seem to get it again later in life. So if they took some of the material from a scab of smallpox and infected the children with that, they found that they were protected. And then this later spread to Europe and actually to America, but there is obviously a very high risk associated with it, and so it was stopped. Edward Jenner in the late 1700s finally did figure out a way to more safely vaccinate against people. And then in the late 1800s, Louis Pasteur developed another vaccine. And so we have been progressing from that point on. What we have learned is that when vaccines are administered, the individual will develop their own, and those antibodies will provide protection. Today, we have multiple vaccines that are routinely given to individuals. Now, some vaccines may only be reserved for certain personnel, such as military, for certain diseases that hopefully the general public may not come in contact with. A couple of vaccines that are shown with these charts here deal with diseases for which these vaccines were developed, and then there has been this very effective immunization program. Both of these vaccines were developed in the 50s, and then we started seeing widespread administration of them in the 60s. The top graph in red shows for polio. There actually are two different polio vaccines that were developed relatively in a very short time frame of each other. And as you can see from the graph, the number of cases dramatically decreased. And as you can see, it's been relatively flat. However, keep in mind that this is number of cases in thousands. One thing that this does not show is that this is just in the United States. Unfortunately, there has been a little bit of an increase in the number of polio in the last several years. Part of the reason for that is because there are some individuals who are choosing not to vaccinate. Usually polio vaccine is given to children. They're choosing not to vaccinate their children because they are under the misconception that polio no longer exists. Yes, it does. And we're starting to see a rise, because if you don't vaccinate your child, you are not providing them that protection against the disease. And unfortunately, we are also, just due to timing, the length of time that has passed since this very effective immunization program was started, we now are having parents who they themselves were vaccinated, who quite frankly have never seen the horrible effects of individuals who are suffering from polio. They have never seen people in what was called the iron lungs, where polio causes paralysis, and if it hits, say, the diaphragm, then you can no longer breathe. And so you had to be put, they refer to them as iron lungs. You had to be put on ventilators to do the breathing for you. Sometimes the effects of polio were milder, where it was very, comparison to paralysis of the diaphragm was considered mild, but still might leave you with crippling effects. Measles, the bottom graph, the vaccine for that was also developed in the mid to late 60s, and as you can see, there was decrease. There's been some little blips of increases, but overall has decreased since the time prior to the general use of the vaccine. Now, unfortunately, this does not show the complete time frame. In 2019, there was a huge outbreak in the United States of measles that could be directly attributed to individuals who had not been vaccinated. Measles is very, very contagious. And so if you have a community where people have not been vaccinated and you have one person who contracts the disease, the others are going to get sick. It is extremely contagious. We base everything else on the measles. So overall, vaccinations have been very, very effective. Now, one concern is that it takes money to develop the vaccines. It takes time to develop the vaccines here in the United States with our healthcare system. We are able to provide vaccines for most individuals. In a lot of developing countries, that is not true. And so that has been an ongoing problem as to how do you develop and administer vaccines to everyone globally. Some vaccines we haven't been able to develop, or some pathogens we have not been able to develop an appropriate vaccine for yet. It just depends. You have to look at each pathogen individually. For some vaccines, with the pathogen themselves, there's high risk, and how each pathogen you have to look at separately as to how you're going to develop the vaccine. Is it a safe vaccine as well? I will say, before we continue further on this, that in the 80s there was a report that came out that stated some possible side effects of being vaccinated. It became very popular. The information from that article was disseminated throughout the general population and popular media forms. And so there's a widespread notion that from this one article that some of the preservatives, etc., in the vaccines could cause autism. And because of this, a lot of individuals made the decision not to vaccinate their children, not to vaccinate with any type of the current vaccines, because they were concerned about these side effects. In the scientific community, this raised alarm because we know the protection that vaccines can provide. And a lot of scientists tried to repeat the results of this particular paper that had been published. No one has been able to repeat those results to show that, yes, there are adverse or harmful effects of being vaccinated, that it could cause autism. No one has been able to repeat that. Several years ago, the scientist that published that original paper finally admitted that all of his work was fraud. He simply made up the data. Unfortunately, that information has not been disseminated widely through social media, and so a lot of people still believe in that fraudulent data. So I just want to stress that the information that the vaccines can themselves cause things such as autism that have all of these lists of harmful effects, that has been shown to be fraudulent data. So active immunization. How do you develop the vaccine? Why are there different types of vaccines? Initially, when early stages of the vaccination programs, they used what they called attenuated vaccines. Attenuated means that it's living, but it's been modified. Basically, what you've taken is that pathogen and you have exposed it typically to chemicals to reduce the virulence. So reducing virulence so it's not as pathogenic is called attenuation. You may still get a mild infection, but it does stimulate a very strong immune response, of the adaptive immune response. One drawback of this is that in someone whose immune system might be slightly compromised and susceptible individuals, it could cause those individuals. So over time, they've kind of switched to other types of vaccines, such as killed or inactivated vaccines. These are obviously safer than the live because the pathogen has been killed, so it cannot cause the disease. Sometimes they may use what we call the whole agent vaccine. You're using the whole bacteria, the whole virus, the whole whatever the microorganism is. One very common thing now to try to use is subunit vaccines. And that's where instead of taking the entire, say, bacteria, you're just taking the antigen, just the protein, and proteins work best. The protein marker, which acts as the antigen that's sticking out of the surface of the cell wall of that bacteria, that's what's generating your antibody response to it. So just take the portion you need. It's kind of, if you're going to respond to, let's say if you're looking at a sweater or a jacket as the bacteria, if all you need is the pocket, don't use the whole jacket, just use the pocket. And that's enough to stimulate the immune response. Sometimes with these inactivated types, you may have to give multiple doses. So you start with one, and depending on the vaccine, it may be a few months later, it may be 6 months later, you give a booster. You give, there are some where you need to give a series of 3 shots. Sometimes they will contain chemicals that will help increase the effectiveness. Toxoid vaccines, where you are giving a modified toxin to help stimulate the immune response. So it just depends on which vaccine you're looking at as to what type it is. A combination vaccine is when it contains antigens from several different pathogens, such as the MMR, measles, mumps, rubella. Research is looking at how do we make vaccines more effective, how do we make them cheaper, and how do we make them safer. And one thing certainly that they're looking at is using recombinant DNA techniques from this means of trying to improve the efficiency of the vaccines. And so this is showing a table for how we're using the recombinant DNA. What are the components of the vaccine? How do we produce them? If you're using an attenuated live vaccine, like I said, attenuated is modified. There are advantages to those in that it does tend to confer lifelong immunity. It does trigger or activate both the humoral and the cell-mediated immune defense systems. The disadvantage of using an attenuated live vaccine, though, is that, as I said earlier, it does put an immunocompromised patient at risk of potentially developing the disease, and there's always the concern that this modified organism could revert back to the virulent strain. So with active immunization programs, we mass produce the vaccines. For bacteria, you can grow the bacteria in very large vats, very large vessels to be able to produce enough to make the vaccine. Now, viruses are going to be a little more difficult because viruses you have to grow with the host cell. They have to reproduce only inside the host cell. So the easiest way of mass producing the viruses to make vaccines against viruses is to culture or grow those viruses inside chicken eggs. The drawback for this is that there are some individuals who are allergic to eggs. If you've ever noticed, when you go in to receive a vaccine, they'll often, one of the questions before you receive it, they will ask you, are you allergic to eggs? And if you've ever wondered, what do eggs have to do with me getting a vaccine? It's because if it is a vaccine against a virus, most likely that virus was grown in an egg. If you're allergic to eggs, you cannot receive the vaccine then because you're going to have an allergic reaction. This is the CDC, Center for Disease Control's recommendation for the immunization schedule. Now, this is from 2015. It is showing from childhood through adolescent and up to adults. I just show this as a reference point of some of the different vaccines that we do have, such as Hep B for hepatitis B, diphtheria, polio, influenza, measles, mumps, rubella, chickenpox, which is the varicella zoster. I am not going to ask that you memorize this because it changes a lot. The tetanus one, I remember for a while it was for boosters every 10 years, then they went to every 5 years, then they went back to 10, then they went to 7. It's constantly changing, and to be honest with you, by the time you guys finish nursing school or whichever type of program you are going through, it's probably going to change again. And so just be aware of it. If you are working in a clinical setting where you would be administering vaccines, say you're working in a pediatric office, you're going to have the most current chart of when to administer the initial vaccine and when boosters are due. These are some of the main vaccines that are currently used. It shows the vaccine, what the disease is, what type of vaccine it is, and how it's administered. And this is a continuation of it. So in terms of the safety of the vaccines, obviously you want them to be as safe as possible. There is always very low risk of a person reacting to it, potentially going into shock. Like I said, there were those allegations that some vaccines did cause other issues such as autism, asthma, etc. But as I said, that has now turned out to be fraudulent. Author of the paper has admitted that it was fraudulent data. Passive immunotherapy, this is where you're administering antibodies that somebody else made. It will give immediate protection for the patient. It's short-term protection. There will be no memory response, but it will provide protection for right now. From taking the antibodies from an individual and then sterilizing, providing those for a second individual, research is now looking at, can we use hybridomas instead, where you take cells from an individual. You have plasma cells basically from a mouse that has been injected with the antigen, so the plasma cells have already produced the antibodies. You then remove those plasma cells, mix the antibodies with the myeloma cells, and have those produce the monoclonal antibodies, just those antibodies for that particular pathogen. This still ongoing research. So the red line on this graph is showing the antibody concentration, and that is from active immunization. That is when the patient or the individual has either come in direct contact with the antigen, been exposed to the antigen, they had the disease, or they received the vaccine. So they are actively making their own antibodies, and as you can see, you have the initial inoculum and then potential boosters later. Either they've been exposed to it again or they got a booster shot, and you can see the level of antibodies continues to rise. The blue line is passive immunotherapy. This is where the individual received antibodies made by someone else. As I said a little bit ago, it gives short-term protection, but not long-term protection. So this is cases of a newborn who is breastfeeding. The newborn is not making the antibodies, the mother is, and the newborn is getting those antibodies through the breast milk. It's giving protection right now, but not long-term. Or where someone is actually injected with preformed antibodies. Once again, the blue lines you can see, it gives concentration of the antibodies right now, but it's going to decrease over time. You'll notice on the axis, the antibody concentration, we refer to this as a titer. I know a lot of you, as you are trying to get into various programs in the healthcare field, they may ask you for your antibody titers. And if you're wondering what they're talking about, it's where you are tested and they actually measure what is the concentration of your antibodies against measles or against hepatitis, etc., whatever the specific one they're asking you for. So a titer just means the antibody concentration. There's lots of different tests that can be used to test for the antibodies, test for the antigens. Serology is just referring to testing the blood serum. And like I say, you may be testing for the antibodies or for the antigen or for both. An idea of has the person been exposed to a particular pathogen? Have they already started to make antibodies against it? So there's lots of different tests. One of the simplest is a precipitation test, and that's where you're going to mix the antigen and antibody, and if they're in a certain proportion, they will precipitate out, and you can see this. Immunodiffusion is a precipitation test. So this just shows that if you have antibody excess or antigen excess, you won't get any type of precipitation. But if you have those two components in the right proportion, the right ratio, then you do see this precipitate form, and you can visually see that either on a plate or on these plates. On the upper zone, there is a well, the blue, where they put the antigen, and then on the right hand side in the red wells where they put antibodies. Both of those will diffuse through the agar on that plate, on that petri dish, and where they meet, you get this precipitation line, and you can visually just see this line. Additional tests are things such as turbometric, where you're measuring the amount of light that passes through a solution, or the methodology test, which is going to measure the light reflected. You can quantify the amount of protein in the serum at that point. Agglutination test is very similar to precipitation. The difference is precipitation is dealing with soluble molecules. Agglutination deals with insoluble molecules. So once again, you can easily see this, so it's easy to read. Hemagglutination is when you're working with red blood cells. This is something you guys saw when you have done blood typing tests, is that you add the blood sample to a slide, then you add what we call an anti-antibody mix. Essentially what you're seeing is agglutination. You see this clumping that will occur. If you have, in this case for blood typing, The bottom slide shows that there is clumping that has occurred in that well that looks yellow with the red clumps. That is where the anti-B antibody was added to that blood. The anti-B antibody bound to the B marker that was on the cell. This essentially indicates that this person is type B blood and it's positive. So you can use this for determining. With the agglutination test, this is how you can measure through titration. You can measure the levels. When you do this, typically you dilute the sample out and you see what's the highest dilution where you still get a positive reaction. So as you can see here on this tray, you had initially your tray, the 1-to-1 is not diluted at all, and then you dilute it out. So 1 to 10 dilution between each well, and it shows listed what the final dilution, total dilution is. And then you add your sample and you look for the agglutination. So on this one, where you see the clumping is at the 1 to 1,000. You can also do a neutralization test where you're mixing. This is often used with viruses. You mix the virus in the serum, you add it to a cell culture, and then you look to see what the is on that. You can also use viral hemagglutination inhibition test. This is often used to detect antibodies against influenza, flu, measles, mumps. The complement fixation test can also be used to detect antibodies. It used to be used fairly frequently because it could detect very small amounts of the antibody that maybe the agglutination test would not pick up. There are typically are done now. Sometimes they will label the antibody, put a marker on it so it's easier to see. The antibody. You could also label antigens. When we label them, oftentimes what we're talking about is adding a fluorescent dye. This can be done either directly or indirectly. And then you look at it microscopically, and if it glows with that fluorescence, then that's positive. You know that it is. So this is just showing how you can add the antigen on a slide. You add the patient's serum. If the antibody is there, it will bind to the antigen. And then you add a label or dye that's going to bind to the antibody. And either way, it's like putting a flashing light on it so you can see it. Another type of test that's often used is called the ELISA test, which stands for enzyme-linked immunosorbent assay. And basically, what you're doing is typically you're looking for the presence of antibodies in an individual's serum. There are different ways that you can do this. Bottom line is you're going to put the antigen in, typically you use these 96 well plates. So there's these little wells, you put the antigen. You can store that antigen for use later, so you don't have to do it immediately. Typically then a protein is going to be added to cover the rest of the well where the antigen is not. And then you add the patient's serum. You add, after that, another enzyme. And then when you add a substrate, if that enzyme is present, it will cause a. And so an advantage of the ELISA is you can use it to detect either the antigen or the antibody. It's very sensitive. You can quantify the amount, say, of antibody. It's easy to perform. You can run lots of tests at one time, so it doesn't take up a lot of space. It's inexpensive. You can actually automate the test. You can prepare them for later. And so there's a couple of ways. So this is what ultimately it looks like. You're looking for that color change. So as long as you have the color change, that is a positive for the antibodies being present. And then the degree of darkness, in this case of the blue color, the darker the blue, the higher the presence there. And so, like I said, this is automated and it can read and quantify the amount for you. You can also do what's known as a Western blot. You've already seen a Northern blot deals with DNA, Southern blot is RNA, and a Western blot is measuring. So you look, each one of those bands is indicative of different proteins that you can. Oftentimes what you're going to be looking for if you're, say, working in a clinic, you want some type of an assay that gives a result really quick. You don't want to have to wait 24 hours. You need to know now so you can decide on a treatment plan for the patient. So point of care testing is where you can do a test. It's very quick, and I'm talking about in minutes. So then you can proceed on the patient. These are similar to ELISA tests, but instead of running it in a plate, it's on a membrane, it's on paper, and you can run it very fast. What happens is there is going to be, excuse me, typically an antigen or the antibody, depending on what test you're running, that is embedded on a paper, on a strip, and then you put that in a solution from the patient, and if the patient has the antibody, it's going to run up, and when you get that antigen antibody complex formed, it's going, you have a dye there where it's going to form a visible line that you can see. This is what they do for like pregnancy testing. This is what they do for some rapid testing for certain types of diseases that they can test it very quickly, like a strep test, for to see if you have strep infection. And if you have the antibodies against it, it forms that very visible line. So I say the idea is you may want to do more confirmation tests later, but this is a very quick way right now to test. This table shows some of the various tests that we have and what they are used currently for testing on these.